DNA is long and double-stranded, right? And while RNA is shorter, okay? And then lastly, it has a phosphate group, okay? So if we lay them side by side and compare them, DNA and RNA, you can see the DNA, if we just look at the strands, right? DNA is double-stranded, it's helical, and it's very long. RNA is single-stranded and it's much shorter. Okay, that's just by strands. If we think about the sugar, then DNA has deoxyribose, but RNA has ribose. And then if we think about the nucleotides that make up the actual molecule, in DNA, it's adenine, thymine, guanine, and cytosine, right? And in RNA, it's adenine, uracil, guanine, and cytosine. And the way you will have to know the base pairings, and you have to know how the structures pair up and what they look like and how to identify them. But the easiest way to remember that is the preposition at for DNA. If you remember the preposition at, adenine always binds to thymine, then by a process of elimination, guanine always binds to cytosine. Okay?
Or they kind of look like each other.
They kind of what?
The C and the G.
They like each other, right? Oh, yeah. So you can see also that I'm representing the A's and the T's with these two little dashes that are lines that are little dashed, right? And I'm representing G and C with three of those. What do those little dashes represent, Anna? Somebody's playing maracas or something, I don't know. Hydrogen bonds, that's right. So adenine, thymine is held together by 2 hydrogen bonds, and guanine and cytosine is held together by 3 hydrogen bonds. Remember, hydrogen bonds single by themselves, not very strong, but a bunch of them together make it very, very strong. And we've already talked about the fact that DNA is very stable in the environment for a long period of time, right? Decades, 50 years, 50 plus years, okay? So now we need to continue on to talk about these things, right? So let's talk about DNA first. The way DNA is constructed is that it's constructed in a double helix, right? So if you think about it, it looks like a spiral staircase, right? And the rules are that the molecules have to fit within that helix. And the only way it's gonna fit in there if a bigger molecule binds to a smaller molecule, right? So let's talk about what those big molecules and those smaller molecules are, right? We call them purines and pyrimidines. The purines are adenine and guanine. They have 2 nitrogenous rings. Nitrogenous rings means that they're rings that have nitrogen in them, okay? And so if we think about them, the nitrogenous rings are adenine and guanine. I am not going to ask you to be able to identify the structure just by looking at them, but you will have to identify them in a DNA molecule, but I'm gonna show you how to do that, okay? The other thing is the pyrimidines, and the pyrimidines only contain 1 nitrogenous ring, and in DNA, it's cytosine and thymine. But there is no thymine in RNA, it's replaced by uracil, okay? Good, so some people have the mnemonic, cut the pie. So cut the pie means C, cytosine, U, uracil, T, thymine, cut the pie, P-Y, pyrimidine. So cytosine, uracil, thymine are pyrimidines. Cut the pie, okay? But of course pie is not spelled P-I-E, it's P-Y, okay? Good, that's pretty cool mnemonic to think about. So here, if we look at the structure, right? We can look at the structure and you can already see how things are pairing up, right? Tell me again what the little bitty dotted lines or dashed lines represent. Hydrogen bonds, right? So here you can see adenine binding to thymine and then guanine binding to cytosine. The way I always remember that is the preposition at. If you remember the preposition at, you know that adenine always binds to thymine in DNA, therefore the other two have to bind to each other in order to meet the parameters and the specificity of the DNA molecule, where you have to have one larger molecule bind to a smaller molecule. It's the only way that they'll fit within that DNA double helix, because if it's too big, it's not gonna work. If it's too small, it's not gonna work. It has to fit perfectly in that actual structure, that double helix. Yes, Anna?
Another way to remember it is apple tree and chew, right?
Dale, tell me what apple tree means.
That AT
Chewy. Oh.
apple apple tree and chewing gum.
Thank you, I learned something new
I learned
today.
that in high
1 point for everybody because Anna made me happy today, okay? So who's keeping them track of points?
Oh, really?
I'll do it.
Okay, you got one.
You've never, this is the first time you said that in this class.
Oh,
Yeah,
really?
yeah, you've never said.
We need a person who keeps the ledger. I guess Sarah's a volunteer. Sarah's honest, she'll keep it right. Okay, so you got 1 point on exam poo number 2. So whatever you get, I'm gonna tack on 1.
Okay.
And if you study with somebody else or with Myron's SI sessions, you get 2 more. Just 1 time. You can't go 5 sessions and get 10 points. It doesn't work that way. 1 time. Okay? My hearing aids are bothering me. I don't even think they work. Yeah.
My ears get itchy with that.
My wife says they work, so I wear them. But I don't think they work. Anyway, okay, so let's talk about this structure right here. So there's a lot going on here, right? So if you look at this structure right here that I'm blocking in, this entire long structure here, that's called the backbone of the DNA molecule. It provides structural support for the DNA molecule. What holds it together are the hydrogen bonds between the 2 nucleotides, right? Either adenine, thymine, or guanine, cytosine. But what provides its support is the backbone. The backbone is composed of the sugar, which is deoxyribose, and a phosphate. Okay? So I want you to realize this little 5 tilde. You see the little 5 tilde? We don't call it 5 tilde, we call it 5 prime. And we call it 3 prime, right? And so I'm gonna talk about those in a minute and explain how we get those and what that means, right? But DNA runs in the forward direction of from 5 prime to 3 prime. Yes? Okay, from 5 prime to 3 prime. So let's continue to talk about this, right? So there is a rule of base pairings, as I told you about already. In DNA, adenine always binds to thymine, guanine always binds to cytosine, right? There's never gonna be an A bind to a C or an A bind to a G. Not gonna happen, okay? So if we think about that for a moment, then, right, the AT molecule, the AT pairing is held together by 2 hydrogen bonds, and the GC is held together by 3. That makes the GC much stronger than the AT. Okay, so if there's gonna be things go wrong with the DNA molecule, it's gonna go wrong with the AT. Okay? So let's talk about the central dogma theory. And the central dogma theory has 2 tenets, right? So central dogma, it's our central law of the way DNA works in all living things. There are 2 tenets, or 2, if you will, 2 statements. The first one, DNA is the master molecule. It controls everything that goes on in the cell. Number 2, under the direction of DNA, RNA makes proteins. Okay? That's the central dogma theory. So how do we put that into context? Well, what we basically say is there's a flow of information within DNA. So within the nucleic acid. So there's DNA to RNA to proteins, right? And so this is described by this beautiful image right here. Here is a bacterial cell, and that bacterial cell has become 2 bacterial cells, but you can see that each of those bacterial cells have all of the components that make up the cell, including the DNA, right? So the DNA gets replicated, and then it gets transferred. One of the newly developed strands goes with one cell, and the other goes with the other cell, right? So that's DNA replication. That's the way that the cells happen. In us, when you talk about mitosis, people always say it's about DNA division. It's not. It's about the distribution of genetic material, because the DNA is already copied and divided. It's just how it's being distributed with mitosis, right? So we're not going to cover that here because that's beyond the scope of the course, and you'd have to take an anatomy and physiology course or a cellular and molecular course to get that information, right? So I'm not going to cover it here. The other one, this way, is about gene expression. It's about how proteins are made. So DNA gets copied by RNA in RNA language, and then RNA is used to make proteins. This other thing over here is really important because recombinant technology, or the making of new organisms, happens in nature, Rebecca, all the time. We just never pay attention to it. But we now have the technology that we can actually create organisms by manipulating them, by taking DNA that are target segments of DNA that are really important and then putting them in other organisms so that we have an organism that's really great. So I would prefer that than others because we can feed the world that way because we make GMOs. So GMOs, if we think about a GMO, genetically modified organism, it happens naturally, but we also have the power to do that ourselves. I would rather have a tomato or an onion that's so big I need both hands to hold it instead of the naturally occurring ones that are this small. You need 20 or 30 of them to eat in a salad where you can cut up one big tomato and take a quarter of it and put in your salad, and it looks good. So, yes, Vivian. Okay. Rebecca, did you have a question? Okay. I love GMOs. There's a lot of people who don't understand them and therefore are afraid of them. They think if you eat GMOs, it's going to alter your DNA and then you're going to be forever changed. No, but viruses do that all the time, yet nobody is afraid of viruses. Right? This doesn't make sense to me at all. Right? And so we go on with our lives. I like GMOs. We have the technology to make them now. We make a lot of them. A&M is one of the leading research groups that has made a whole bunch of genetically modified organisms for our betterment. Right? They've actually made a bonnet that is maroon color, and one year when they were still playing the University of Texas and they don't like each other, they planted a whole bunch of seeds on the UT campus and they all bloomed maroon, and then they said, oh, we did it, right? And so it was kind of a big diss to the UT folks. Yes, ma'am?
So are we looking at binary fission?
That is correct. We are looking at binary fission.
So transcription and translation still happen with binary fission?
Yes, ma'am. So transcription and translation just talks about how DNA is replicated. It doesn't talk about the distribution. So it doesn't talk about the phases of mitosis, right? That's a different process. The phases of mitosis deal with how DNA is distributed to the new cells, but the DNA has already been replicated by then. Okay? That's a great question. Anybody else have questions? Okay. So there is a really cool thing that happens in some viruses, like the retroviruses, like HIV or feline leukemia or things like that, where the flow of information is usually DNA to RNA to proteins, with here the RNA is the base. And so they have to use an enzyme that makes DNA from RNA, which is reverse. So we call that reverse transcriptase because now they're using RNA as a template to make DNA, right? And that's pretty cool. And it only happens in a few viruses that we call retroviruses because they use reverse transcriptase. It'll be on the exam. So if it says which of the microbiological entities represents reverse transcriptase, it's going to be HIV. The answer is going to be HIV. Okay.
You said retroviruses.
Retroviruses. Mm-hmm. Retroviruses. And retroviruses, it doesn't mean from the 50s, right? If you dress retro, right? This doesn't mean that at all. It just means it's reversed. So when we talk about, we're about to be talking about DNA replication in bacteria because they're easy to talk about. They don't have a lot of chromosomes. They have one, right? Where we have 46 in total, right? And with us, all the DNA is being copied at the same time, so it's really hard to, what's going on, right? It's all this stuff going on. When we can talk about bacteria, they have one chromosome, they have one site of origination, and that's it. So they're really easy to talk about, right? And so we're going to talk about that. Replication of the DNA molecule, whether you're a prokaryotic or a eukaryotic cell, is enzyme mediated, right? So there are some enzymes I'm going to hold you responsible for on the exam on Tuesday. And we're going to talk about those in just a second. Let's talk about semi-conservative replication. So semi-conservative replication is the idea that when the DNA molecule gets replicated, when the DNA molecule gets opened up, because it's a helical structure, when it gets opened up by enzymes to get copied, each of those strands we call the original strand. Some people call it, Christine, the parent strand. Some people just say it's the old strand. Each of those strands, the original strand, is used as a template, and then a new strand is made for each of those, right? And now you have two new DNA molecules, each representing one original strand from the original DNA and a new strand that was made from that strand, using it as a pattern, okay? That's better illustrated by this particular image. So here you have the original parent strand, and it opens up and it gets copied, but it gets copied so that there's a new strand that is added to each of the original strands when the DNA gets opened up, right? It's called semi-conservative replication because half of the DNA gets conserved in each of the new strands that is being copied, okay? And now think about that for a moment. Why is that important for any organism that has to have its DNA replicated in order to continue to survive, right? Well, the answer is, well, maybe, do you know the answer? What's the answer? What do you think? Jasmine? She's like, hell, don't call me Proby. Yvonne? She's not even looking at me. Vivian? Huh? Why is it important that one of the original strands gets conserved when the new DNA strands are being made? Christine, do you have a hypothesis?
Survival?
Not survival, but if you have, that's a good guess. If you have, yes.
Mutations.
What about mutations?
The new strand, if it didn't keep the same original strand, it could have mutations which
That is
could
correct.
alter the DNA.
So if you have the original strand being conserved in each of the DNA molecules, it's less likely that there's going to be mistakes, right? That's pretty good. Polymerase, which is the enzyme that copies the DNA.
Survival is a good answer then.
No,
Oh,
no,
no?
no, it's not a good answer.
It is, okay,
Well, argue with me. Tell me why it's a good answer.
because that's what viruses, bacteria, what
The viruses don't work the same way.
organisms are all trying to do. They're all trying to
Correct.
survive.
Correct. But what you want is
And
more.
genetic mutations can mess with that. I'm sure there's.
In a negative way?
it could, in
Hold
a
on. In a negative, because I want her to.
That's right.
Okay, sorry.
Both, it can, some things will adapt right to the environment.
No, adaptation doesn't work that way. Adaptation doesn't work that way.
Mm.
Okay? But what you can have is a mutation, but most of the mutations in us are bad. Yeah?
But for viruses, it's not.
Oh,
Not all the
true,
time.
but the viruses act
They're
very
trying to
differently.
survive.
But they don't act this way. They don't do this.
Hmm.
Okay? So the answer is so that there's less mistakes or there's no mistakes. It's not foolproof, right? There's like 0.1% of having a mistake in the DNA that's being replicated. And usually in us and in bacteria, there are mechanisms that can correct that. In bacteria, if it doesn't correct, then they're mutated. But in us, if it doesn't get corrected, it becomes a bad thing. What does it become? Cancer. So why isn't cancer destroyed by our immune system? Ruth? Donna?
Isn't it often?
Huh?
Isn't it often? Doesn't our immune system?
It doesn't. Huh? No? Why isn't cancer destroyed in us? So cancer can be in us 20, 30 years before it becomes symptomatic.
Wasn't it because there's a mutation in the cells that get it to keep growing, and then that one mutation gene is keeping it.
That's true, but why doesn't our immune system attack it?
Because it's our body.
It's
Because it's our cells. And it's labeled us. So the immune system if you don't want me to touch you, tell me. Our immune system comes over here and goes, but you're labeled correctly. I'm gonna leave you alone. Right? But Christine, to your point, we're now working on technology that we can add a little bit of DNA that changes the outer structure of our cell. And now our DNA will say, you don't belong here. We're gonna destroy you. Oh, wouldn't that be cool? In my lifetime, I've got maybe 20 good years left before I croak. In my lifetime, I think we're gonna have a cure for cancer.
I don't understand why we don't.
Well,
pretty sure we do.
by idea, by concept, we do. But it has to go through clinical trials before we can even use it. Now, don't get me wrong. You know, when people have stage 4 cancer and they say there's nothing we can do for you, I would ask, if it was me, is there a clinical trial that I can become a part of? And then they say, well, here's 3 of them. I'm gonna look at them and say, this one looks like it might help me, so I'll do this one. And if I die during the clinical trial, at least I've added to the body of science that will help somebody else. But maybe I get cured with a process that's not accepted. Yes, sir.
My grandmother had the second most aggressive form of cancer. It's some sort of thyroid cancer. Most thyroid cancer is good, not good.
And easily controlled, yeah.
But there's one that is like anaplastic thyroid cancer that has like less than a 1% survival rate, and when they diagnose it, you usually have less than a month left. Went on a clinical trial and was alive for 3.5 years after.
Yeah.
So
when you have cancer, it's not about how long you're gonna live. It's about the quality of life you're gonna have while you're still alive. Okay? So that's the important thing. So if I can live 6 months and be really happy and do really cool things that I wanna do, or if I can live 1 year but be in pain and not be able to do anything else the entire time, but I'm still gonna die, give me those 6 months. Right? Yeah. That's what happened with my dad. My dad, I told, he was stage 4. I said, Dad, what do you want to do? Here are the options. He said, I want to go around and visit my friends and say goodbye to them. I said, okay, I can do that. So I was going to meet with him every Friday, take him to other cities so he could say goodbye to his friends. And I left for the weekend. I went back on Monday, and my mom and my sister got a hold of him. And when we talked again, he said, oh no, I want to fight it. And the last 6 months of his life were horrendous. He lost all his hair. He couldn't eat. They stuck a tube inside so it went into his intestinal tract. He was a big man, and he lost probably 130 pounds. So he was at about 102 pounds when he passed. He was nothing, and he was depressed the entire time. What kind of life is that? Yeah. And he was giving things away, right? He was giving everything he cared about away to other family members, right? Yeah. So to me, that was not good. And I told my mother and my sister that, are you sure you want to do this? Because it's going to be very hard for him. And they were very selfish. They said, yes, we want to do this. We want him to have a chance to survive. I said, but you understand his chance of survival is less than 20%. He said, we still want to try. And then we saw the horrors of what happens when somebody is terminal with stage 4 cancer. I have a lot more to the story. If you want to hear it, I'll be happy to tell you about it. But I'm not going to continue on here. Okay? Questions? And I can talk about it now without getting teary-eyed because I've processed it a lot. Okay? So let's talk about the steps of replication, right? So it's enzyme mediated. What does that mean, son? What does that mean? It's enzyme mediated. Means that enzymes control the entire thing. So usually DNA is tightly wound up like this. It's if you will, it's uptight. It's just tightly wound up. So in order for it to be replicated, for those of you doing Levaquin, listen, because it's part of what you're doing. There's an enzyme that comes over, and this is all tightly wound up. An enzyme comes over called topoisomerase. Topoisomerase comes in, and it relaxes the DNA. So it's no longer uptight, right? And then there's another enzyme that comes in. It's called helicase, and helicase opens up the DNA molecule. So now it's ready to be replicated. One strand is called a lagging strand, the other strand is called a leading strand. But polymerase, which does the copying, doesn't know what to start. So there's another enzyme that comes in called primase. And primase puts a little bit of a primer, a little bit of an RNA molecule in each of the sections that have to be copied. So now polymerase knows when to actually copy. So that happens in both the leading and the lagging strand. The lagging strand is a little bit different because the leading strand, if we think about it, the leading strand is copied continuously from a 5' to a 3' direction. But the orientation of the lagging strand is backwards. So it has to be copied backwards. So in order for that to happen, because of the limitations of polymerase and the way polymerase can work, it has to be cut into pieces. We call those things Okazaki fragments. And so what happens now is primer has to come in and it has to put a piece in between each of those fragments so the polymerase knows where to copy, and then it gets put back together again. The thing that nicks them or cuts them up is called an endonuclease or a nuclease, and the thing that puts them back together once they've been copied is called ligase, okay? So there are 6 enzymes I'm gonna need you to know for the exam, and they are topoisomerase, helicase, primase, and polymerase. There are different types of polymerases, but for the scope of this course, I'm not gonna expect you to know the difference between polymerase 1 and polymerase 3. Then you'll need to know for the lagging strand, the nuclease or endonuclease, and then ligase, okay?
What's that last word you said?
Huh?
The last word you said?
Ligase.
Oh, like-
Mhm. Okay? Good?
What's the poly, what was the second sentence?
Polymerase.
Yeah, that's how you spell it.
There was something underneath that, I thought, no?
What's that?
Underneath polymerase, is there something underneath?
Yeah.
Oh,
This-
lag.
this-
They will lag.
This was, I rewrote it because it was kind of skimpy looking down there. Okay?
Oh wait, could you? Okay.
There's
6 total.
Does that say endonuclease?
So nuclease and endonuclease are the same thing. People just use those words interchangeably.
Do we need to know the Okazaki?
Yes.
Yes. Okazaki fragments are the little individual segments on the lagging phase, and the lagging strand. Okay, so. Again, here, if we look at this, it's 5' to 3'. And let's talk about this. Let's talk about the structure of the DNA molecule. So, Daniel, I need you. So Daniel and I, come stand here. Daniel and I are driving a car. Get your wheel ready.
Wait,
We're driving a
Why
car.
are we both driving?
We're in 2 different cars. So we're driving down the highway. We're driving parallel to each other. Go stand over there. Now, DNA runs anti-parallel. So when we run anti-parallel, we're driving towards each other. So we're driving, this is anti-parallel. DNA runs anti-parallel. Okay? Thank you, sir. Let's give him a hand because he's a good trooper. Okay, good. So if we think about that for a moment, we say that the forward direction is 5' to 3'. Okay? What that means, it's going from a phosphate ending to a hydroxyl ending. So that little tilde is prime. And what that means is that that phosphate group is attached to the 5th carbon of the sugar. So let's count them. This is the first one. So everywhere there's a point, there's a carbon, unless there's some other atom at that point, right? So you can see there's an oxygen there, right? So here's a carbon, that's 1, 2, 3, 4, and there's the 5th one. So the phosphate group is attached to the 5th carbon. It's on the 5' location for that sugar, yes? So that's the 5' end. The 5' end always has phosphate, okay? The 3' end has hydroxyl. So now if we come down to the bottom and count the carbons, 1, 2, 3, at the 3rd carbon, at the 3' location, there's a hydroxyl. And now you can understand how DNA is being replicated, because DNA always gets replicated from 5' to 3'. Okay? Yes? All right, now, can you appreciate the middle of these things, right? So we talked about the sugar and the phosphate being the backbone, giving the DNA structure. But what allows the DNA to come together and bind to each other or be attracted to each other are hydrogen bonds. Can you appreciate that some of these pairings, right, have either 3 hydrogen bonds or 2, right? So therefore you could tell which of the base pairings those are based on how many hydrogen bonds there are. So if there are 3 hydrogen bonds, what's the base pairing? Guanine, cytosine. If it's 2 hydrogen bonds, what's the base pairing? Adenine, thymine. But now I even want you to be able to tell me which nucleotide is there. So now you have to figure out which one is the pyrimidine and which one is the purine. Yes? So if we look here, if we look at this base pairing, how many hydrogen bonds are there?
2.
2. Therefore, what base pairing is this? Adenine, thymine. Now, which one is the pyrimidine and which one is the purine? The pyrimidine is a small one, right? And here it would be this one. And so what is that? Thymine. Therefore, that has to be adenine. Here is how many hydrogen bonds here? 3. So that has to be which pairing? Guanine and cytosine. Which one is the purine? Guanine. So which one, this one or that one, is the purine? That one, that's this one over here, right? Therefore, that one has to be cytosine. Will you be able to do that for me on an examining tool?
Yes.
You'll need to be able to do that. Okay?
So the purines are bigger or the?
Purines are bigger. Cut the pie. Those are the small ones. Yes? Good? All right. So, here are the enzymes that you're, let's see. No. Not, well, yeah, there's one missing. So, did everybody get a copy of, those are the only ones I'm gonna ask you about, the ones that were on the board, nothing else. Because it's very complicated, I'll just make it very easy to understand. Okay? So, let's look here for a minute. Okay, this is a really great image. So can you tell me what is represented by 1? What's being represented by 1? Which one?
A start codon.
No, this is not protein synthesis. So it can't be a codon. Kimmy.
Would you say this is one of your favorite images?
No, one of my favorite images. If it's gonna be on the exam, right? How about if I send you all the answers later? Yeah, but let's talk about it now. Good? So this is an enzyme that relaxes the molecule. What is it? Topoisomerase. This is an enzyme that opens up the molecule. What is it? Helicase. So in order for the DNA to be copied, it has to have a start place. The pink, the light pink, is the enzyme that puts the starting section into the DNA. Primase. But the pink is the actual starting part, and we call that a primer. Okay? Good? So, if we think about this for a minute, endonucleases are not being represented, but the ligase is, right? And so here is the ligase, which is actually annealing. It's putting the image back together again. I don't have an A and a B, but I probably will have an A and a B on the exam. This up here might be A, and if it's A, this particular strand is being copied continuously. What strand is it? The leading strand. The other one is being cut into pieces, and that is the lagging strand. And this is actually a piece of that DNA that's being copied. And what do we call that piece? An Okazaki fragment, named after a very famous scientist who came up with the idea of the way that the lagging strand was replicated. So if we think about this for a minute, the leading strand is copied continuously, all at once, no stopping, about 1,000 nucleotides per second. We can't even appreciate how fast that is. The other strand, because it's of its orientation, It has to be cut into pieces, then copied individually, then put back together. Okay? So I know I've said a lot, and usually probably, I have not sent it to you. People say probably, some people say, do you have a video that we can watch? I do have a video. It's my favorite video. And so, hey Sarah, will you get the lights for me? I want you to appreciate the beauty of the way the DNA gets replicated, because it is absolutely fascinating. Here we go.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Absolutely fascinating. It's kind of funny because if you show YouTube, you can always tell what the interest of the person who's showing you because they show you things that probably I don't want you to know about me, but okay. Like I'm a big basketball fan, right? So not necessarily an orange guy fan, right? So
Why?
No way.
I have a weird question.
but
Has there been medication developed that can target the lagging strand? Because I feel like it's more vulnerable.
that's a really interesting thing. I don't know the answer to that, but what I will tell you is we have medications now that can target the individual mutation and it can fix it. Or it can send a signal to the cells that have that mutation so that the signal is received by the cell in us and those cells die. That's called apoptosis. And so those are both really good medications. Both of them are used to treat cancer. Again, I think in my lifetime, we're gonna have a cure for cancer, right? But is it gonna be affordable? That's the thing, right? So that's the problem with the way we're going. To me, I'm gonna get in trouble. I'm gonna say it anyway. To me, healthcare should be affordable for everybody. For everybody, not just the people who have extra money. That's not the way it is. Okay. Any questions about DNA replication? Okay. So let's talk about protein synthesis. Right, with protein synthesis, the same enzymes are involved, except I'm not gonna go over all that stuff again. Right, all I'm gonna do is I'm gonna start where protein synthesis actually begins, right? And so if we think about that, remember the 3 different types of RNA that I said we were gonna talk about because they're really important. There are like 22 different types. We're not gonna talk about all the types, right? That's for 1406. But for here, we're just gonna talk about messenger RNA, ribosomal RNA, transfer RNA, right? And so if we think about the process, this is the DNA molecule, and this is RNA coming in and making a copy of it in RNA language. What that means, Rebecca, is that instead of the T's, they're gonna be U's. Because protein synthesis will only work if there are uracil in the molecule. So that's a good way to tell them apart, is if they have T's there, DNA, if they have U's there, RNA. So if we think about that for a moment, then that process of copying DNA into RNA is called transcription, right? It's being copied. Just like I'm talking here and hopefully you're writing notes, and when you're writing notes, you're taking what I'm saying and you're writing it down in a manner that it makes sense to you, right? That's transcription. The other part of it, the actual use of the information in the messenger RNA by transfer RNA to bring amino acids to the actual protein that's being made, and then the ribosome kind of folding up that protein is called translation, okay? So if we think about that for a minute, transcription is the job of messenger RNA, translation is the job of transfer RNA, and the folding of the protein to make it unique and make it work, because what establishes the functionality of a protein? Its shape. So once it's folded up and has shape, it becomes active. That's the job of the ribosome, to fold it up. Yes? Then it gets released. Okay? So if we think about this for a minute, here it is. I'm going to talk about it in order so that we can make sense of it, but it's happening at random and all at once. It's just a crazy mechanism, right? It's beautiful, but it's crazy. And to some people that look at this, it can be overwhelming, right? That's why I'm going to talk to you so that it's in order, and then you can really appreciate what's going on. So let's talk about the parts, right? This is in a eukaryotic cell. This is the nucleus, there's the DNA, this is the RNA, this is the ribosome, right, attached to the endoplasmic reticulum, right? And so you can see that once the messenger RNA is made, using DNA as a template, it copies it, it leaves the nucleus by a nuclear pore, and it forms a complex with the ribosome. Now, if we look at this message, oh, you scared me for a moment. I'm sorry. Sarah, I should know that by now, but I'm sorry. I didn't mean to. But anyway, so here you can see that in the messenger RNA, there are a series of nucleotides, right? And each 3 in a series is called a codon. How many people have heard that before? A codon is equal to 3 nucleotides, and the 3 nucleotides code for 1 amino acid. Yes? I have given you all a codon table, right? Did you get one, Fatima? Anybody else missing a codon table? Thank you. Okay. You do not need to memorize this table. I'm going to give you a copy for the exam. I'm going to give you a copy. You're going to have your own copy for the exam. You don't have to bring it. I'll give you a copy. Okay? But each of those particular 3 nucleotides is 1 codon. And the reason that this little firecracker looking dude, that's the transfer RNA. The reason the transfer RNA can bring in the exact amino acid that's coded by in the messenger RNA is because in the transfer RNA, there's an anticodon. And the anticodon is at the base of the transfer RNA, and it is complementary to the messenger RNA codon. Yes? Are you with me? Yeah. The codon is in the messenger RNA. It's 3 nucleotides. And the reason, Vivian, that the transfer RNA knows exactly which amino acid to put into that series is because the transfer RNA has a anticodon that is complementary to the codon in the messenger RNA. What does that mean, complementary? If the codon is A,
It matches.
the anticodon will be,
Oh, T.
no,
Oh, wait. Sorry.
it's an RNA, it'll be
Oh,
U,
U.
right? Are you with me? If it's G in the codon, the anticodon will be
C.
And if it's C in the codon, the anticodon will be
G.
Therefore, there's almost no mistakes. I mean, there can be mistakes, mutations, right? But we're not going to talk too much about that. It's a little bit outside the scope of the course. Okay? My job is to give you the basics. tRNA, the anticodon belongs to transfer RNA. Okay? So, the protein synthesis happens in 3 steps. There's an initiation, an elongation, and a termination, right? So the initiation is what starts protein synthesis. And that initiation sequence is what we call a start codon. And the start codon is always going to be represented by AUG. And hence, because I'm not here to make the test harder just because I can, if protein synthesis is going to start, it's going to start with AUG with me. Okay? Good? I'm not going to put a bunch of them before and then stick AUG, and then so that's where you have to say it starts right there, right? Good? So once it starts, then as the messenger RNA moves through the ribosome and the transfer RNA reads individual codons, it's going to bring individual amino acids to bind to or attach to the beginning chain of the peptide or the polypeptide. And you're going to have amino acids being attached to amino acids, right? That process is called elongation. So here you can see that elongation occurs at the site of the ribosome, and you can see that there's a chain of amino acids, right? So let me come back here for just a minute. There are 3 sites within the ribosome. There's an E site, a P site, and an A site. How many people heard this before? The A site is the acceptor. That's where the anticodon is accepted by the ribosome to actually attach to the codon. There's a P site. The P site stands for peptidyl. Peptidyl means to put into a chain, right? To put into a chain. Think of peptidoglycan, right? There's chains of sugars. Here, the peptidyl is a chain of amino acids, yes? And then there's an E site, which is the exit site, where the actual transfer RNA is released, okay? Hence, you have this long chain of amino acids, okay? Until there's a termination sequence that occurs. The termination sequences, there are 3 of them. They're UAA, UAG, and UGA. If any one of those shows up, they don't code for an amino acid, they code for basically stop, okay? And at that point, protein synthesis is terminated. It's done with. Okay? Ma'am?
Does that leave a tail? Like unpaired? Does that
It's
leave a tail
unfair.
that's
It is
unpaired?
unfair. Okay? Alright. So we know what a codon is. We know what an anticodon is. We know they're found. Right? And so how do we know which amino acids go in a sequence? Well, because we have a lot of research and therefore we have a lot of theory, and that theory has led to the development of a codon table. This is the codon table, right? So this is my favorite codon table, and this is what you'll get on the exam. You'll have this for your use. You can also bring a calculator if you want, okay? But I'll show you why you don't really need one, but if you want one that makes you happy, you can bring one in, okay? So if the way you read this is the first base in the codon is here, then you take the second base from here, then you bring the third base over, and wherever those 3 things come together and intersect, that's gonna be what amino acid you have, okay? So for instance, ACC, not Austin Community College, but the codon ACC codes for what amino acid? ACC, threonine. Now you'll notice that a lot of these things, all 4 of them, no matter, like if you have CC, you can have CCU, CCC, CCA, CCG. That last nucleotide can be anything, but it all codes for the same amino acid. We call that the wobble, because it doesn't make a difference what that last nucleotide is, they're all gonna code for the same amino acid, okay? Wobble. You're gonna see that on the exam, be sure you remember it. Wobble, okay?
How do you spell that? Just
W-O-B-B-L-E, wobble. Like the Weeble wobble. Yeah, wobble.
No matter what the last.
Doesn't make a difference what the last nucleotide is, they all code for the same amino acid. Yes, ma'am?
What is it?
The wobble,
Where? Like on there?
It's being represented here, but it's just the last nucleotide. Can you see right here? This is CCU, but it can also be CCC, or it can be CCA or CCG, but they all code for proline. That last one, it doesn't make a difference because they're all going to code for the same amino acid. Okay, good. So
in here, it is this is the one.
then they
and
don't
this
follow
is-
that rule.
Oh,
Okay, they don't follow that rule.
So,
Okay.
But you just ask what is wobble, then we just understand what.
And
it is
you
and
can just say, in most cases, the last nucleotide doesn't change the amino acid. Okay? All right, so GGU stands for what? Use your codon, tell me.
GGU.
Glycine. Very good. What about ACC? Oh, you already did this one. 3 What about AT or TAG?
Wait, TAG?
Kimmy? Why isn't it on there, Kimmy? That is exactly right. There are no T's in RNA. So I'm just checking your total knowledge. I'm not trying to be tricky, Yvonne. I'm not trying to be tricky at all. Okay? So, all right. What about this one? Could you give me the complementary, this is DNA, could you give me the complementary RNA strand?
A, U,
Go ahead. G, what?
C,
CUA, what?
U, A,
Mm-hmm.
G, A, U, G, A.
There it is. Good. So I'm going to ask you to do this some way on the exam, right? And so if we think about this, what does AUG code for?
Start.
Huh? The start. Don't tell me start. Tell me what amino acid. Methionine. What about CUA? Leucine. What about GGA? Huh?
Glycine.
Glycine. What about AUU?
Isoleucine.
And UGA. So you'll need to be able to do something like that on the exam. Okay? Any questions? Okay. Okay. So, Sarah, will you get the light? This particular video works. It's a little bit more monotone, but the information it is portraying is really good.
Translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formylmethionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formylmethionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Any questions?
So elongation is the amino acids binding to make a polypeptide chain.
Correct.
To a protein.
It can be a polypeptide, but it can also be a peptide. Right, peptide is less than 50 amino acids, polypeptide is 50 or greater. Ruth, did you have a question? Anybody else? So we move into the last topic for this particular unit, which is mutations. There's a lot of stuff up here, but the easiest way to remember a mutation is that a mutation is a change in the DNA structure that is inherited, okay? Or inheritable, because the cells can give it to whatever cell comes after it. So it doesn't really, the mutation really doesn't have to be at the gamete formation. It can be there, but it can also be with the somatic cells, right? Because all of our somatic cells can have mutations in them. Most of the time, they don't get through checkpoints, they get destroyed or they get repaired. But if they get through checkpoints, then these cells that are not our normal type of cell can go unchecked for a long time, and they can become, or they will be cancer. Right? People can have cancer for 20 or 30 years before they start to have symptoms. 20% of us in this class will be touched by cancer in some format. 20%. Okay? So if we think about that for a minute, let's talk about mutagens and epigenetics. So epigenetics is the study of how the environment can change our cells. And so there's a lot of things that are out there that are mutagenic. My father developed gastric cancer, and I believe that his cancer was actually caused by a cat tower explosion in Corpus Christi. Does everybody know what I'm talking about? So there are these big refineries in Corpus. One of the refinery towers blew up. And there was all this poisonous, toxic stuff that was released in the atmosphere. And when that happened, the people who were living in that area could not leave because of all of the emergency units coming in and out of there, right? Either taking people to be helped or coming in to try to stop the fire. So a lot of those people were sequestered in that area. So my father was sequestered in that area for a day. He couldn't leave. And he was an insurance salesman, and he would go over and he would collect money from people to pay the insurance, right? But all the people in that neighborhood got a lot of cancer in Corpus, including my father. He didn't live there, but he was exposed for a day to all that noxious stuff. Now, I don't have any real proof, and when I brought it up to the state epidemiologist, they were not allowed to study it at all. I wonder why. Because the oil companies down there are very powerful, and they didn't want to know what the results of those studies were, because if you know the results of the studies, you have to do something, right? So that study never got done. But yet, a lot of people my father's age who were in that neighborhood had cancer and all kinds of cancer. Yep.
Is that why the water is really nasty?
In Corpus?
I went last year and.
So, what do you mean the water's really nasty?
It has a lot of tar.
It's poopy?
Tar.
It's what?
A lot of tar.
Okay, but not the water you're drinking,
No, the beach water.
the beach water, there's a lot of tar in the beach because of all the oil that's being drilled offshore. And so it's less dense than the water, so it gets washed up on the,
So all those signs about naturally occurring, blah, 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 that's not real. I mean, I thought it was BS, but.
it does naturally occur, but there's too much of it to be natural.
Yeah, there was chunks of it all over.
Be sure you take baby lotion when you go down there the fall, 2 weeks from now, right? Because if you take baby lotion, you can take that tar off your feet very easily because slime dissolves muck. Don't try to get it off with water and sand, it won't work, but baby oil will take it right off. Or you can buy that, like down there in those little shops that you can go in that are tourist traps. You know, they'll sell you the tar removing substance for like $8 or $9. Or you can buy baby oil for like $1.50 or something like that. And it works just as good. Make a lot of money off of you all who don't understand science. Okay? Good? Be careful of the Portuguese man of war. This is their time of year. So if you're going down there, if you see, you know, everybody know what a Portuguese man of war looks like? It's a jellyfish that's blue, has like a little crest on it, but that's just the actual floating device. The tentacles are all in the water and they can extend by 10 feet. So if those things wrap around you, you're gonna be in pain because that's some of the most severe pain I've ever had when I got wrapped up with one of those guys. And the only reason I got wrapped up in one of those is because my little cousin went in there and thought it was a balloon, and he picked it up and it wrapped around him and it stung the hell out of him. And I went in there to grab him and I grabbed him, and then the jellyfish got all on me, so I was in pain. But the little kid was he fainted. He was in so much pain. We took him to the hospital. Those things are not to be messed with. And if you pull them, like usually if I see them, I'll pull them out on the beach and then I'll wrap them up and I'll throw them away because they're gonna hurt somebody. But do not step where the tentacles were because the toxins are there also. The venom is there, and so it'll hurt you. Yes,
Do you know like the MOA of putting meat tenderizer on a jellyfish leg?
So it will actually denature the venom, and it'll help it so it doesn't hurt as much. You can actually put a little bit of acid on it too, but to cover it with sand. And when I say acid, I don't mean like hydrochloric. I mean like vinegar. Some homies down in Corpus carry a little beach going pack, and they have baby oil and meat tenderizer and things like that. But that denatures the venom and it doesn't hurt. It doesn't work for everybody though. Yeah. It's better just to stay here in Austin and just go to Rebecca's bar and drink a little coffee and play pickleball.
There you go.
We'll get a people out there.
Okay. So let's talk about how genes can be transferred to organisms, right? So bacteria are some of the few organisms that can take up DNA from the environment. They can just take it up from the environment. And when they do that, they can incorporate it into their own chromosome. That's pretty cool to think about how that happens. But it can happen lots of different ways, right? So that's one way, right? There's a very famous study that there was a person, Griffith, he was a researcher, and what he did was Klebsiella pneumoniae produces a capsule which helps it be very dangerous to us. He had one that produced a capsule and he had one that didn't produce a capsule, right? He called it the smooth form that produced the capsule. He called it the rough form that didn't produce the capsule. And so what he did was he killed all the organisms that were the ones that had the capsule, and then he took that substance and he mixed it in with the ones that didn't have the capsule. And the ones that didn't have the capsule, the rough form, took up the DNA from the environment, from the broth. And then they incorporated that into their own DNA, and they started to produce capsules, right? So this is a way that shows that bacteria can take up DNA from the environment. Isn't that cool? Okay. So transposons are little pieces of DNA. They can be plasmid, they can be lots of different parts, Britt. But if we think about that, then these things can actually not jump, but they can be put into the chromosome. So we call them jumping genes, transposons, real small pieces of DNA. They can become part of the chromosome. Okay? And then lastly, you know about conjugation, and you know that bacteria can transfer plasmids from one to another, right? And what's the appendage they used to do that with? A pili, right? And so these plasmids can also become transposonic. They can actually be put into the chromosome, right? Remember, bacteria don't have a way to have sex. So they can't really make new organisms, where if humans have sex and they have a baby, that baby is a accumulation of half of the genes from the father and half of the genes from the mother, and that particular baby or that particular individual is unique, and there'll never be another one of them. So you are the only one, Anna. There'll never be another Anna like you. There'll never be another person like you. My cousin used to tell me when we were younger, in the world somewhere you have a twin. I'm like, what? I'm like, what? He said, somewhere in the world you have a twin. I'm like, oh, that's not possible. I said, my mom and dad had me. They'd never been anywhere. There ain't nobody like me anywhere else. So she looked at me, she said, you're such a nerd. And then she left me alone. She thought that for me forever. And now she calls me for advice all the time. My son is sick, surrounded with him, what happens? And I'm not a doctor. But you know what's happening, tell me. And so I'll tell them. And then she goes to a doctor, gets a prescription. She says, you're right, this is what it was. I'm like, I know my stuff.
If you clone someone, it's the same thing.
Yes, they would look exactly like you, that have the same physiology, but it wouldn't be you, because you brought up, and the things that influenced your life are your parents, your and your values. Genetically and physiologically exactly alike, but different, right? But you know, there's some people who are saying that perhaps we should clone an individual for spare parts. I'm like, okay, I don't believe in that. But why don't we just take stem cells, and if you need a new liver, we can make a new liver for you. Why do we have to create a whole organism? Why do we have to keep a clone?
For fun. To torture them so they have feelings. I'm sorry.
You are so bad.
I mean, we do it to people.
The last way is transduction, and transduction is delivery of a nucleic acid by a virus. And so this can happen in us, and it does, right? So think of herpes or shingles, where with herpes, you know, you get infected, and then that's a gift for life, right? And so you have herpes forever. You can't get rid of herpes. We don't have the mechanism, we don't have the understanding of how to get rid of herpes. So if you got it for life, it's for life. And periodically you're gonna have episodes of herpes showing up. And that can either be genital, but it can also be buccal cavity, right? So fever blisters, we call them cold sores, we call those things.
Doesn't like a very high percent of people?
Yeah. And so they usually show up when people are stressed out, right? So like, you know, when there's a lot of exams happening, just look at people walking down the hall, and you're gonna see people with it. And here's the interesting thing. When spring break is over, when people come back, watch how many people have cold sores. Because the sun is a stress to the body, and if they already have herpes simplex 1 in their body, it's gonna show up. Watch how many people come back and have cold sores.
Why is it some people it lie dormant and some people?
It'll lie dormant in everybody, but at some point, the stress is just a lot. Because those viruses are kind of interesting. They mostly affect you when you're at your lowest point.
If someone is like pregnant and has herpes when they give birth as a baby?
Not necessarily. We have ways to stop that. Right now, there have been cases where babies have been born with herpes, but they don't usually survive. They die.
Would they be having, like, would the person who's pregnant have to have like an outbreak of this or something?
Yes. But we have really good medication that can control that. So there's no reason for somebody who has been infected with herpes to not think that they can't have children.
I have friends who have multiple kids and they're married and they've never given it to their partner and
There's lots of ways.
their partner doesn't
You can
have
only
it.
give it to somebody if you're having an episode.
You just have to know,
Yeah.
you have to pay.
I don't know, somebody over here?
Where do cold sores come from?
from somebody else. So if you have a significant other who's got a cold sore, do not kiss that significant other when they have a cold sore, because then you'll acquire it. Okay? Just say no.
Why does everybody have HSV1? Just because
It's pretty
the
prevalent.
grandma kisses the baby and
Yeah,
the baby gets
yeah,
it or whatever?
yeah. I don't have it. I mean, I've never had a cold sore in my entire life, right? But a lot of people do. My wife doesn't have it either. But a lot
One
of
time
people do.
I had an old roommate, and I used to raise her, and I freaked out, whatever, I went to the doctor, I'm like, Haley has it, and she's like, I'm not gonna test you for that. Everyone gets it, and it's like.
A lot, yeah, cold sores are very common.
Yeah, they were like, it's a waste of the test, like, it's not a big deal, and I was like,
Yeah.
man, I was very young, and didn't know anything. about
Shingles
it.
is much more serious.
Yeah.
So you get chickenpox as a young person, and then it's dormant forever. And then at some point in your life, you get really stressed out, and it develops along the nervous system of the body. So usually people get it down their neck or across their back, where all the major nerves innervate.
Interesting.
Oh yeah.
Shingles is contagious?
A couple of years ago.
But only by the pustules. I mean, so think about this. Chickenpox is contagious how?
Right. You have to touch.
So if somebody has shingles, you can get it through the air. My Aunt Chema, the curandera, her son got chickenpox when he was really young, and she invited us over for a party. And we went all to the party. We were just like little stupid kids walking into the chickenpox den. And today we're gonna have a party for my cousin. I'm just gonna name him Daniel. And we're gonna have a party for Daniel. And we were like, it's not Daniel's birthday. We didn't know why we were having a party, but we had a party, it was in the summer, so that we could all get chickenpox at the same time and then not have to deal with it during the school year.
Yeah, parents still do that.
Yeah.
It's
Why?
hard.
Why, is there a difference in having, now kids don't really get it because they all get vaccinated, right?
That's correct. But if you get it as a young child, it's less serious.
Yeah, if you have it as an adult, you can,
It could
cause really bad complications.
If you get shingles as an adult, if you get chickenpox as an adult, it can be very serious.
My doctor was telling
Yeah.
me about it. He's like, I want you to get it.
It's always better to get it as a young, or get vaccinated. But there's so many people who would rather lick a lollipop or get their kid to lick a lollipop that has chickenpox and then give it to other people in the family so they can get it. That is the silliest thing I've ever heard of. When you can take a vaccine and get the same protection. Having that is called a no-sode. People also do it with snookers and they rub it into like here. Right?
So it's the same protection, is it the same? Or
You get the infection. Oh, look, somebody's blowing their nose out.
You didn't get one side versus the other, one of them's not stronger or better or less longer.
It's all the same. It's just how it affects you. Right. Yep.
I have a question. It's just from what I've seen. For HSV, it's only transmitted when you have a flare-up, right?
HIV?
Or, no, HSV herpes?
Yes, only when you have a flare.
Okay, because I had a patient who came in panicking because she had an outbreak for the first time in her life. And then she was accusing her boyfriend, and she was with her boyfriend for 2 years. So I was concerned that I'm still, and I'm supposed to.
So
Did you get that from her?
you
Had that
could have got it.
before?
You could have got it from the boyfriend, but the boyfriend would have also had to bring up with that. They would have had to have had an event. And really, pro these rules of engagement are what?
If it looks nice,
Check-
reasonable, don't touch it.
Check out the goods before you engage, right? And you're like, looks okay, then go ahead. But if it looks funny, if there's open lesions, or if it smells bad, get the hell out and get out quick.
I thought with herpes you can still get it if they're in the early stage, like.
Only if there's an open lesion. But how else are you gonna get
There
it?
has to be a lesion?
Because how else are you gonna get it? If you're not being exposed to it, if it's still in the body, you can't really get it. It has to have some, but sometimes the lesions can be hidden and you don't know. The person might know because they have a little bit of pain, but it's not noticeable, right? So you just have to be careful. There was I don't know, every once in a while I get these Reddit things that are kind of funny, and one of them said, I have a good friend who has developed a relationship, and his partner said that before they could have sex, they needed to both go get checked out for sexually transmitted infections, right? What do you think about this? And like everybody was like, oh, it's not good. I'm like, actually, it's really good. It's very responsible of two individuals going together to the doctor, getting checked out, coming back negative, and do the hell what you want after that. But if one of them tests positive, then you make a decision, right?
But it's an informed decision.
Correctamundo.
Yeah, and like if someone argues with me, but I'm just like, obviously, we don't wanna go business with me, you don't wanna be with me, with me. That's just how I feel about it.
But you gotta be careful with the Latino men. Why? Because they'll look at you and they go, "Ay, mi cielo, te quiero con todo mi corazón," and they'll like really make you feel, "Oh my God, he really loves me." And don't fall for it.
You think the white dudes won't do that? Come on.
But they don't use the romantic language of Spanish.
No.
It's a different thing. Oh yeah. So it's pretty interesting to think about, right? So I make a little bit of fun and have a little bit of fun when I'm talking, but really it's pretty serious to think about, right? I want you all to be healthy. I want you all to be safe, especially as we get closer to spring break and some of you are going to be headed down where there's going to be 100,000 other individuals and hormones are raging. Take care of yourself. Yep.
So about the chickenpox party, would that be acquired immunity or herd immunity?
Both. And it depends, because you'd have acquired immunity because you come down with the infection, but herd immunity because you become part of the vast network of people who actually had or been exposed to or had a vaccine for chickenpox. So therefore, there's limited spread in the population.
We can have both.
Nice question.
Is it, would you say it's more beneficial to get it, I know you said there's no real difference, you still have the same immunity, but with vaccines, say, I don't know, if there's other things in them, that could potentially be harmful, so wouldn't you rather go?
So
like, further
vaccines
away?
are biological by nature.
Yeah.
One out of every 100,000 individuals are going to have an anaphylactic reaction to a vaccine.
Yeah.
Statistic. But if you give it to 1 million people, 1 million people are protected, but only 10 of them develop a reaction.
So do people ever have an anaphylactic reaction to get it? Get a bunch of kids together, all with it, and then you say, technically, it's like everybody got it, as 3 of your kids now.
Yeah, but I would prefer, because there can be so many other things that happen with infectious agents like that. You could have damage to different organs. You could have, if you're with a child and don't know it, you can have birth defects. There's a lot of things that can happen that you can complete control of those vaccines.
Yeah.
So I would always take the vaccine. And I've had more vaccines than the average bear because of what I do. Right? I'm still pretty normal. At least I think I am. So, other than that, I'm a big proponent for vaccines, but I also look at data, right? So the COVID vaccine, to me, the best vaccine was Pfizer. The other two, Moderna was good too. J&J, I'm a J&J boy. That's my company. I worked for them for a long time. But the J&J vaccine was horrendous. It was terrible. I'm embarrassed that they even put it on the market. It had a 74% efficacy. The others had 90%. So I won't talk about it when we talk about vaccines later this semester. Why don't y'all take five minutes and come back, and then we'll do the review, because I'm done, right? Exami Poo on Tuesday, all multiple choice, with the exception of your presentation. We'll review when you come back. And then we go to lab. Yeah. Okay, so 5 minutes and then come back.